scientific poster and you're set up around the room and you're answering questions, doing that part. You may be a more behind the scenes role where you're compiling all the data ready for the presentation groups. So you might want to start thinking about what group you want to be in. Your teacher will be giving you probably the options to choose those a little bit later. But start thinking, do I want to be the person up front talking to everybody else? Do I want to be a more behind the scenes person? I love glitter glue more than anything in the world, and I want to be on the poster group. Keep in mind it's like a scientific poster, not super fancy glittery one, although that's okay. So anyway, just something to start thinking about. And again, if you're taking good notes today, you should get a lot of good information to start your research papers. So that's all I really want to go over now. Your teachers will be talking about more of the details about the actual project. Just don't forget that this site exists. All your rubrics are on there. All of the stuff on what to do for a presentation, what to do for your paper, it's all there. So just don't forget that it exists. A lot of people do. And I just wanted to make sure you are aware of it now. If you do go to the Governor's School website, I'll show it to you. So BRVGS. And if you go to current projects, it's right here, Arabidopsis research project with Virginia Tech. So you can get to it through the Governor's School website. You can just Google it like I do, and that's all you need. So any questions before we get started? Thanks, I appreciate that, Michael. So, all right. So without any more speaking by me, I'd like to introduce you to our contact person from Virginia Tech. We've been working with him for many years now. So David Lally, it's all yours.
It's really great to see you all today. Everyone's intelligent and alive and awake. I really like working with the Blue Ridge Virtual Governor's School. For one, I have inordinate affection for humans in general, but I really like this program. I can almost promise you that the things that you learn in this program will be some of the most valuable things, because BRVGS was an idea, can we do this? Can we make this thing happen? So you're learning communication skills and collaboration skills, bringing you all together from these different schools, and creativity, all the big important things that you really will need in the workplace. So I'm really happy to work with you guys again. So my name is David Lally. I want to say right from the start, please feel comfortable asking me questions at any point. That's not just today. You can just stop me, call out, raise your hand. And but during the experiment, if you have a technology for communication, I probably have access to it. I have my cell phone number. If you need to FaceTime me, whatever you want. You call me in the middle of the night with some issue, I probably will even answer, but let's try to keep it silent. I was in high school too. There's me. Nice weather here. So I can relate. I have 2 teens at home. I'm an advisor for teens. I understand. So the whole idea, the big goal, is to bring that research from Virginia Tech out to high school classrooms. I work with high school classrooms around the state, and all of the scientists I work with are working with this plant called Arabidopsis, Arabidopsis thaliana. And it's a tool, it's a model organism that they use to study genes and genomics. So those scientists send out mutant and wild type seeds to the classes. Does anyone know what a mutant is? Yeah, it's genetically modified, right? Changed in some way, either by just happened in nature, or we have intentionally changed it because we have that ability now. How about wild type? Anyone know what Yeah, it's the control in genetics, right? If you change, in science we do the same thing over and over again. We change something, and then we compare it to the original to examine the consequences of that change. So you guys will have the mutant and the wild type seeds. You're going to grow them under experimental conditions that you help design, and then you're going to hopefully let the scientists know. You'll be working with a scientist at Virginia Tech. She's the department head, so she doesn't get out much anymore, but her senior research scientist will probably come for your presentations. She has it on her schedule, so they're always very interested to see what sort of things that you found out with their research. So I always like to back up a little bit, because people forget about how valuable plants are. And I ask, why study plants? What benefits do we get from plants? Way into that.
Oxygen.
Good, oxygen. Clothes. We got a great deal of fibers from plants, right? What other ways do we use plant fibers? Anyone know?
Medicine.
Okay, medicines. So we get a lot of chemicals from plants. Food. We got food. The fibers. Plants make an amazing assortment of chemicals. There's at least 200,000 chemicals that we know of that plants make that aren't directly involved in the plant's primary metabolism. They call them secondary metabolites or natural products. So we're very interested in those. We have a drug discovery center at Virginia Tech where scientists are investigating extracts from plants from around the world to discover new drugs. Because those chemicals are very interesting. A lot of them are active on humans. And as somebody said, pharmaceuticals, about 30 to 50% of our pharmaceuticals come straight from plants. The fibers we use in a great, when I say fibers, does anyone know what particular molecule that I'm thinking of? It does have carbon. Well, it has carbon atoms. Okay, we're scaling up. We started with carbon. Now we're on glucose. No, that's a good drug, but I'm thinking about the fibers. So next level of organization, what do you do with all those glucose subunits if you start bringing them together? Cellulose, it's a very, very valuable molecule, the most abundant organic molecule on the planet. So whether it's this stuff, cotton, we wear it, we write on it on our paper, we get a great many benefits from cellulose. Anything else you can think of? What else are plants really good at? Those green plants?
Habitats.
That's true, habitat. We all have habitats. Animals need habitats. Pretty much everybody's going to use, some wood at some point, unless you have a steel and glass house. Furniture. So they soak up carbon dioxide from the atmosphere, and then what are they doing that for? What are they making it for?
Their energy.
Glucose, which is what for us? For them. Energy, right? So plants are the brokers of the sun's energy. All living things are open systems. That means we take in energy and information from our surroundings, from the environment, right? We all need energy to survive. And we are not very good. In fact, you could sit me in the sun all day. If you don't feed me, I'm going to be a pretty sad guy. We can't broker that radiant, that light energy and take it from the sun. We need plants to do that. They have that ability. So fuels, we use it for, food is fuel for our body, but we are also using a great many things to supply our energy needs. Can anyone name a plant fuel? Ethanol, we can make our own, right, by fermenting corn. Alcohol, right, we're making alcohol. What else? Anyone have a fireplace?
Yes.
You can just put it in there, right? And add a little energy to start that reaction where oxygen is going to recombine with all those reduced carbon molecules. You know, I'm talking about reduction. You get chemistry yet? Yeah. We'll get there. It's important. So, how about old plant fuels? Ancient stuff.
Oil.
Fossil fuels, right? Where do they come from?
Underground.
Underground. What? How'd they get there?
Plants.
Compacted by sedimentary rock.
All right, wait. Let's compare what I derived from the word salad to individual. Hear what? But what was pressurized?
Plants. Dead plants.
Right.
Dead plants.
Something with a photosynthetic ability. It could have been microbes. Some are almost entirely from plants. I put this picture up here. What kind of plants do you think those are? Ferns. Good. There's a particular kind of fern called a tree fern. You can see that darkness there is actually the trunk. Here I am in a kind of blurry picture. Can't blame my wife. She took a good picture. I was just blurry that day. But there I am standing next to the trunk of that tree fern. They're still around. They're not as big as they once were. But, you know, we do these depictions in textbooks where we try to say, what did the Earth look like about 300,000 years ago? And you'll see that the dominant tree at the time was a tree fern. We don't have any fossil evidence that flowering plants had evolved or even cone-bearing plants. We just had these tree ferns, giant tree ferns. Did what plants do, took the light energy, turned it into that chemical bond energy, got buried, pressurized, and now we can dig it out of the ground as coal. So coal is almost entirely tree ferns that have fallen down and stored for that energy for eons. Okay. So bottom line, can we live without plants?
No.
We're entirely dependent upon plants for our survival. And right now we have somewhere around 7 billion people in the world. They're expecting, anyone know, by about the middle of the century? They used to say 9 or 10 billion, but they just like increased a little bit by the end of the century. I think it's up to maybe like models are predicting maybe 10.4 billion. A lot of them in Africa. So scientists have always played a role. You know, humans make technology in order to satisfy their needs, extend their abilities. So scientists, the big role that they have is to try to solve problems. We've always played a role in trying to address agricultural problems, how to increase food. We're doing a great deal of stuff right now to try to meet the needs of all those new humans as our resources decline. Okay? So it's very important if you're thinking the big picture, what are we doing here? What is all this research about as you're just like measuring little plants and stuff? Why am I doing this? Well, you're doing it because that's what we're interested in right now. We're trying to figure out how plants work at such a fundamental level so that we can make them change in sophisticated ways and meet the needs of humans so that we don't go extinct. So, here's Arabidopsis. It's a lot bigger on this picture than it is in real life. Here's my little scale bar. That's a 2.5-inch pot. It's a little tiny weed. Nobody grows it for anything in particular. Can anyone give me a reason why we chose this in science as a model organism? It grows fast, good.
Very
Sure.
few genes compared to other plants.
Yeah, it's a very small amount of DNA compared to a lot of plants. Plants have hugely different DNA amounts, so that's good. It's what? Yes, it's simple in many ways. You know, for one, it has all the features of a plant, but it doesn't do anything that's doesn't spray poison, that or anything. Interesting. There is some value in that because, see, it's a brassica. Anyone know what a brassica is? Can you name me any other plants? Like
Corn.
no. Radish. Cabbages. Anything else? Anything with that sharp flavor? That mustard,
Mustard.
canola, right? those glucosinolates that like, whoa, you know, cook a Brussels sprout too long, you walk in the house after going to a movie, and you're like, what you know, like, that's in the same family. So here we have versions that haven't been cultivated. So they do that, right? In fact, a lot of our bioinformatics, our computational approaches to biology now are looking at crosslands and comparing it to what we found from this little simple plant with a small genome. We could sequence it very quickly, finish it in the year 2000, and you know, we're learning a lot. So here, basically the two main things. It's physical features. It does grow fast. It is small, and it makes a lot of offspring. If you're studying genes, you're interested in heredity, and if you have a large pool of offspring, it bolsters your confidence in drawing conclusions from the outcomes of those experiments. And then just its genetic features. Not only does it have a very small genome, that it does most of the things that other plants do, a very compact genetic package. It also is very easy to change genetically. So we can make those changes and then examine the outcomes, and it helps us to understand the basis of those genes. What is the connection between the gene and the trait? Okay. Any questions yet? We good? All right. What animal are you? What's that? A wolf? Awesome. So, here's Brenda Winkle. She's your partner scientist. She is also the head of the biology department. And she studies a class of plant chemicals called flavonoids. Has anyone ever heard of a flavonoid before?
Flavor.
Does you have? So, if you look at this slide, with all these images, what's most striking? What's that? The tasty specificity. Yes, anthocyanin. This reddish, purple, blue color, pigmentation is one of the things that flavonoids do. Very good. And anthocyanin is responsible for that. You know, some people like to set up a little experiment with blueberries. If you take the juice out of the blueberry and change the pH, that the color will turn red. So you see that range between red and blue. Okay? So even though you may not be that familiar with the term flavonoid or with the chemistry of flavonoids, you eat them and see them all the time, right? Fruits, flowers, and vegetables in your life. So we are studying those plant chemicals, and we break them into groups. That's why I said it's a class of plant chemicals. Flavonoids are way down here. They're a subgroup of what's called the phenolic compounds. All of them together are those secondary metabolites. Like I said, there's at least 200,000 of those. They usually are classed by their starting material. Terpenoids, you would recognize again, just from your experience, if you smell a nice piney smell in the forest, that's alpha-pinene, that's a terpenoid. It's made from isoprene, which is like a little Lego block. It's like you can make a great variety of structures from the same set of blocks. That same 5-carbon molecule can be arranged into thousands of chemicals. Flavonoids all start with basically an amino acid, phenylalanine, and then the plant has this great ability to make all these different chemicals. So we're really interested in how the plants make these chemicals. If we master that, then we can create our own systems to do very interesting chemistry. So I'm going to give you a little rundown on the kinds of things, the kinds of ways that we have seen flavonoids being used by the plants. Because we really don't know what their role is. So we just look for examples in nature. So we know that there's about at least 4,500 known flavonoids. In Arabidopsis, you'll find about 200. They are traditionally known for their ecological roles. Didn't even know what ecology is. Good, but what specifically about what plants with
Study
what?
of ecosystems.
Ecosystems, but specifically the interactions, right? interactions between organisms and their environments and each other. Okay? How do all those things come together to form that more higher level of order and complexity called ecosystem? First thing I wanted to show is this picture.
That's awesome.
I think it's kind of cool. What do you think's going on there? Can you describe that to me?
Trees growing.
Plant growing? What's that?
It's growing into the shape.
Growing into the shape of the sidewalk, what is it? The roots, right? So you're saying it's not random, that it's following the pattern of the pavement? Why do you think it's doing that? Path of least resistance. What's in the soil? Nutrients and water, right? So could be path of least resistance. Could be going for the water, the nutrients, anything else you can think of? I mean, the tendency of a root, if you look at any plant, you can move it around and you're going to see gravitropism kick in. The roots go down and shoots go up, right? So it's part of that momentum, but it's a little bit more sophisticated here. It could have something to do with avoiding mechanical stress. You can see that it's in the middle of a walkway. I believe it's in Thailand. People walk over it and they scuff it, and the plant might be trying to get away from those little scuffers that live in the above ground world. But the main thing I wanted to point out is the plants move very slow. Any fans of Tolkien and the Lord of the Rings will recognize even Tolkien playing with that idea because the trees vent. How slow it is to decide anything. Well, we'll have to meet on it for a few decades. And we'll talk about it. Plants move much slower, but they are interacting in their environment in very sophisticated ways. In fact, organisms are not just a product of their DNA. A lot of people say, oh, DNA is a blueprint for who we are. But really, it has a lot to do with those. Okay. Here's some examples. We see flavonoids being used as pigment, as I showed you in that slide. Here are 3 different structural diagrams for 3 different flavonoids and the colors that they create in those flowers. And plants want to attract pollinators. It's a very efficient way to have sex. Minimizes the amount of sex cells that they have to make. They give a little trade to some pollinator. Here's a little bit of tastiness or a bad funky smell, whatever. You just had filled the corpse plant bloom at Virginia Tech. It blooms every 5 years. It smells like dead bodies. Everyone celebrates. So I showed you some things that weren't flowers that were very colorful, fruits and vegetables. Why would a plant invest energy to color them?
And then they have a secondary.
Oh, spread the seeds.
Nutrients to grow, but also, what else?
Reproduction.
You know, you're attracted, you go into the supermarket, oh, look how shiny and red that is, right? Carries it away, seed dispersal, good. So you're broadening the range. Broadening the range might mean that, okay, maybe that little niche over there experienced a drought, but over here where I threw that apple core, well, it's not having a drought, and you're increasing its range and increasing its chance of survival, that organism is more fit. If you look at any organism and look at its adaptations, somewhere along the way you're going to come back to reproduction, maybe to sex, because that's why we're here, because we're good at making copies of ourselves. If we're an extant, a living organism, that's what we do. You can also see flavonoids being used that aren't pigments, but they still have, later on in chemistry, you'll learn something about these kinds of, phenyl rings, these bond systems that are very good at absorbing energy without getting damaged. So UV light can be very damaging to us and to other living things. So it's good to have a sunscreen. Flavonoids can serve that role. They can also act as a sort of insecticide. So if the plant is getting chewed on, in this case with sorghum, that plant will produce more of this particular flavonoid and will discourage those insects from chewing any further. You also see examples where flavonoids are used to encourage those other living things in their environment to them. Plants' #1 need for nutrition, mineral nutrient range, is nitrogen. There's abundant nitrogen sitting above us in the atmosphere. However, plants need it in the form of ammonia or nitrate. They get it through the soil from the roots. They can't change that gas, that gaseous nitrogen, into those nitrogen compounds in the soil, but there are microbes that can. So they set up a symbiotic relationship with those nitrogen-fixing organisms. They build them a little home right on their roots. Those microbes make nitrogen compounds in exchange for food and nice happy residence. In order to get those microbes to come to the roots, they send out these two flavonoids through a process called chemotaxis, following the concentration gradient to its strongest point, its strongest at the source, right? Just as if somebody put a plate of hot chocolate chip cookies in a house and blindfolded you and asked you to find it. You think you could find it?
Yes.
Right? We'd use a similar process because you can sense chemicals in the atmosphere too, and you would follow it back to its strongest point and enjoy that moist, warm, chocolatey. It's actually close to lunch to say. So just to sum that up, why we do this kind of research. One, if you want to understand plants, like I said before, plants are obviously making these chemicals. And those abilities themselves, that chemical ability to make all that great variety very specifically, is very interesting to us. So we'd like to understand it for plants alone. But then we found that flavonoids are very good for humans. A diet rich with flavonoids, not straight. You can't snort a line of flavonoids and expect to live for a long time. That's kind of toxic. But in the amount that you find in a diet rich in fruits and vegetables, chocolate. So many articles now about chocolate, dark chocolate is good for you. It's flavonoid content. They have all those great properties of being anti-inflammatory, anti-cancer, antiviral, all of these great things. So we're really interested in how they're made so that we can maximize that experience for humans and improve human nutrition. We good? Good so far? Questions? Comments? I'm moving too fast, too slow?
I think it's good.
Can I move on?
You said something about chemotaxis, something where you go to a strong source. What was that word again?
Chemotaxis. CHEMO. That's the chemical, and then taxis is like a taxi. Right? Taxis. Taxi. I think I'm detecting the greatest note-taking activity from Lisa. Why is that? Is this the new school? How's the new school? That's good? Did it inspire you?
Where does awesome like that?
It is awesome.
I'm being forced.
I know the deal. All right, let's talk about the genetics really quick. What do genes do for you? Anyone tell me? You keep talking about genes.
They're what helps you. They determine what your body needs, what protein you need, what cells need.
Everything about you? You nodded. You nodded something. You have the Louisa group about human and thoughts, chimeras. You know what a chimera is? Anyone nerdy enough to know what a chimera is?
I know what it is.
All right, let's hear it.
Isn't it like they have like the head of a lion,
Yeah,
the
yeah.
tail of a snake?
It's a fusion of two
Percy
different life
Jackson.
forms. What's that?
Percy Jackson.
Ah, look at that. Nice. Movies do have a role.
That's right.
All right, so genes are going to determine what all those things that we talked about. What do you call those? Are phenotype, can we use traits? Because I wrote it on the slide. So I said they determine our traits, but they're really helping, right? I want to look at this. So give me some examples. Here I see like he's talking about heredity. I'm assuming they're related. What do you think their relationship is?
Physical.
No, it's gotta be like.
What's the relationship?
Parental.
You think this is the mother?
No, they look about the same.
Really?
They do look like
This
this.
is the mother-daughter, right? Probably mother-daughter. I am from Boston. Have I cursed you?
Yeah.
I have?
He's about to.
It's hard. It's a discipline. It really is. My wife said to me after visiting somebody last weekend, you know you cursed twice in front of Karen. She's only 11. I was like, I did? Anyways, we have funny accents and colorful language. So, name me some things that presumably the daughter inherited from her mother. What traits? Hair? No. Okay, so sight. Presumably she didn't come equipped with those glasses. That would hurt. Her eyesight, right? Do you think our genes can predispose us to certain ailments or diseases?
Got her nose.
Okay, so we have some physical traits. We got the hair, the nose,
Her chin.
her chin. She got the drop chin.
It's like.
Anything else? Okay, choice of clothing. Trying to get into that next level now. What's the mother saying?
Obnoxious.
Obnoxious, right? She's saying she inherited that. She inherited a behavior. Can we inherit our behavior?
Yep.
Yeah. You can inherit
My mom was
Okay, so the thing I want to point out by making that statement, genes determine our traits. Well, there is another factor that's determining our traits, right? What's that other thing? Our environment, right? So really, when you look at anything, you have to consider the interaction between the environment and with our genetic payload. In this case, she's saying her personality is more obnoxious. Well, it's hard to tell because we have this other level of complexity. We have billions of synapses for every gene we have in our body. It's this whole other beautiful level of complexity. And yet all of those neurons have genes, right? So if we want to convey a message through our brain, we often will do it with neurotransmitters. And those neurotransmitters are coded for by genes. So we see subtle changes in the way people behave. If you bring it over to the plant world, do they have brains? No, but they do make those very interesting set of chemicals, right? So they are interacting with their environment, and if you look at how they respond to things in their environment, many times you'll find, well, they're really manipulating those other organisms. That behavior of the plant looks very intelligent. So it takes stepping back and sort of redefining how we might view intelligence, right? They don't have a brain, and yet their signal response system is adaptive and very sophisticated. We just learned that, when bees are in the hive, honeybees are in the hive, they have this, the larvae there, and there are these nurse bees, and they feed them with this goo from their head called royal jelly. Have you ever heard of royal jelly? Right? So that's sort of like mother's milk. It has a yummy cocktail of all sorts of bodybuilding, chemicals, lipids, and proteins, etc. Now, at a certain point, those female, potentially female embryos, some will go on to become queens, and some will become workers. Okay, so what decides that decision? All of a sudden, the nurse bees with the goo in their heads, remember them? They start giving them pollen, fermented pollen from a plant. Well, they just discovered that the chemical P. coumarin, which incidentally is the starting material for all flavonoids, is very rich, and it goes and it expresses genes in those particular larvae that would make them a queen. So basically, by feeding them a plant chemical, they're limiting the amount of queens that we have. So plants are doing these sort of, that may seem like, unintentional, but the more you look, you start to see, well, bees are a pollinator of plants. So they have this intimate relationship with their pollinators, with humans even. Opiates, they are a plant-made chemical that bind to our receptors. They didn't know why cannabis had its effect on people. Turns out that's how we discovered that we make our very own cannabinoids. Plants are mimicking them, right? Why? Who knows? These are the things that chemical ecologists study. But the bottom line is that you have to look, especially with plants, if you want to understand something about their genes, because all those chemicals are made by proteins, and proteins are coded for by genes. So that's why when you do this experiment, you're going to be changing something in their environment. Okay? We're trying to understand what's the role of all these genes in the plant. And with a plant especially, it's going to have something to do with its genes. Okay? So everybody knows this sort of stuff, right? Genes are made of DNA, and DNA are made of these things. Nucleotides. Yes? This isn't new, right? And then genes code for these things, like I said, proteins. And you can see this is just an example of 6 different proteins. They're not all plant proteins. I think that one in the middle up there, that's a microbial helicase. So very specific nucleotide sequence results in a very specific amino acid sequence, which folds up into a very specific shape and a very specific function. Does anyone know what a helicase does? Good guess. Yes, that word helix is almost in helicase, so it has that ace at the end, which often means it's an enzyme. So the DNA kind of goes through there and separates the 2 strands of DNA. Anytime you copy a cell, you have to copy the DNA, you have to separate those 2 strands, and those will be the template for the new strands. That tiny little psychedelic Cheerio does it at a rate of about 500 nucleotides per second. Very efficient little machine. You may not think about them, like, anyone recognize this protein? It's pretty, isn't it? It's actually 2 different genes, 8 copies of each, so it has 16 subunits. And it's probably one of the most, if you were to grind up leaves from any what we call a C3 plant, which is a lot of plants that we eat, so Arabidopsis is a C3 plant, about 50% of the soluble protein would be this protein, which is rubisco. And this is the protein that stitches those carbon dioxides, those carbons together to make those carbon chains that are higher and that everything else relies on. So we're very interested not just in the plant, but these are nanotechnology wonders. So not only can we borrow from them, but we can also learn how to maybe make them more efficient. Rubisco is horribly inefficient. It binds oxygen, right? So if there's too much oxygen around, it doesn't work that well. We could change that so it only binds carbon dioxide. You see all sorts of strategies, there's other kinds of photosynthesis to try to deal with that because it ruins the molecule. So we're also interested just how these wonderful, beautiful proteins work. They're tiny little machines that do amazing things. Dr. Brenda Winkle, your scientist, is trying to identify all the proteins that are involved in that flavonoid chemical bacteria. A whole bunch of different genes are responsible for making those changes. Starting with that P. coumarin, which gets a coenzyme A stuck on it, and then all the other flavonoids are made from it. Starting from that point, all the other enzymes, those little machines that are working in that chemical factory. She believes the gene that you are working with, the mutant line that you're working with, is involved in making those. She's trying to understand now and validate that that is actually true. So in your mutant, it has been knocked out. Okay? So if there's no AADL gene, and you don't have an AADL protein, if there's no AADL protein, well, somewhere in that chemical factory for flavonoids, you're not going to have a worker. It may be pretty necessary. So you'll see some things change in its flavonoid chemistry. And since flavonoids have a role in how plants interact with the environment, maybe if you grow them under different environments, you will see that they respond differently. Most of the work that Dr. Winkle does, especially since she became department chair, is all biochemical. She doesn't do much of what you're going to do, which is trying to understand how changing the gene affects the phenotype of the plant. Questions? So you're going to be asking that question. Comparing the mutants and wild type, just asking, how does disabling that gene affect the plant's traits?
What kind of traits would it change?
Well, we've grown it for a little while, so we can see some changes that just happen under standard growth conditions. But you can imagine that we see roles where it has an antioxidant role, so it might respond differently to light. You know, certain things that create oxidative conditions like heavy metals and stuff. That's interesting to look at because they tend to create oxidizing conditions. But we don't really understand even just the anthocyanins. We don't fully understand how the plant uses them. The more I work with Arabidopsis, the more I see little things I didn't notice before. So, for example, if you're watching the plant grow, the volt is made, the reproductive structure, and then right where the flower comes out, the little stem is a pedestal, and not a pedestal, pedestal. And what you'll see is right before it flowers, it's a little pink in that discipline. I don't know why. It happens. So anthocyanin, just that one, is being used in all these very sophisticated ways. So bottom line with the scientists I work with, you really like to see the things that students come up with. You want to see your thinking rooted in the biology of the plant. Why would that make a difference for a plant? So I don't want to discourage anyone's to ask students to rethink things if it seems a little too far-fetched or doesn't relate. I've had students ask everything from can I try Viagra on it? It's like, well, why would that be relevant to a plant? If you can explain that to me, that's fine. Sometimes people want to say that change the pH, but they want to use like an organic acid. And plants are very susceptible to organic acids. So when I say organic acids, anything with a carbon molecule, so sulfuric acid, H2SO4, no carbons, that's an inorganic acid, that's fine. But I would use acetic acid, carbonated. I would kill it, probably in one application. So I try to, scientists like to see that you're kind of basing it on the biology of the plant. But some of the things that students find out are kind of accidental. So if the scientist comes out, they'll see it. The students might not even notice it. Students up in Fairfax County and other scientists that I work with, you just introduced this mutant line. It was a double mutant, two different versions of the same gene. And she was seeing this difference in the way the students' plants were growing, apparently under the same set of lights. Well, one shelf, it turns out, great classroom management by the teacher, every student had the same spot across the shelf. They had their name on it. They didn't move it around. Oh, there's less light at the ends of the rack than in the center. That's twice as much, right? So if you looked at the way that the plants were growing, typically, wild type, you can see this little hill. You left them in the same spot. Right? They'd be highest in the middle because they got the most light. Well, the mutants were responding differently. She got on the down, called her lab. This is all this cool thing. See if you can reproduce it. That's the kind of thing. It might not even be something that you intend, but scientists do get benefit from this program. So hopefully, Dr. Hildreth will come and present so that she can gather those. I will try, if I can, to come during your experiments. Feel free to contact me if you want to Skype or look at the plants. I don't know what's going on. Good. So, what's next?
My question is, so you talked about all the different types of things that flavonoids do, and like coloration, pest control, recruitment, and everything like that. Like I know ADL's like background of a lot of things. Do you know like kind of where those paths lead with those different effects yet? Flavonoids.
They've
I
done.
know there's a coloration thing, but like, do we know anything else yet with just to help direct our variables a little bit by other effects?
Well, I mean, you're somewhat limited in the things you can do. You know, having a bunch of insects and stuff is kind of a problem, but you can certainly, continue to try things with light, different chemical treatments, different nutrients is also very interesting. Varying light and nutrients sometimes is interesting because that's how nutrients are built. Flavonoids have so many different roles. It's hard to tell like what specifically.
Is it behind the scenes enough? It could be any of those things, I guess.
Yeah, I mean, like clearly it seems like the roots are lighter color, so they're responding to light.
So is AADL like a flavonoid?
AATL itself.
Like, I mean, it's a protein and a gene and all that stuff, but does it go to be a flavonoid?
So now, a lot of our hypotheses come from data. So if you have an interest in biology and in computing, it's a really great and exploding field. Big data is huge because we have gotten better and better. We have reduced living systems to information. And at the same time, we have developed a technology that is exceptional in computation. So we are learning more and more how to make use, ask questions of those big sets of data, algorithms, and actually get hypotheses. So one thing that scientists do is they expose the plants. You can look at this database, the ePlant database of the University of Toronto. Where is this gene expressed? Which tissues? Which points in development? People have subjected the plants to all different kinds of environmental conditions. That's another level of complexity. If anyone was interested in doing that, that would be great, because that would be a great, way to build a hypothesis. Well, I looked on the ePlant site, and it looked like that the data, that the gene was expressed, at this particular point in development. So that's why I was interested in focusing in on that. So, to answer your original question, you can also say, here's 5 genes that we know from evidence, from experiments, are critically important in making any other genes that turn on and off in the same pattern. So you can do pattern matching. You can do like temporal, time of development. And this gene was pulled out. It behaves just like a flavonoid gene because all the other big flavonoid genes, it's getting turned on and off at the same time. Does that make sense? So it was identified computationally.
Usually, if you had a bigger idea on that, would you have $200 original text on that, or just not?
When you say a big idea, like what? Do you have something in mind?
Yeah.
Okay. Is it a secret? All right. You can tell me afterwards. Depends on what it is. Like if it's something, that you have. Dr. Winkle's lab is not a very rich. She spent more time running the department than writing grades. So a lot of times that answer would be yes. I have to deal with it. All right. Yes?
You were talking about I don't consider that, but you said the ways where the hired. What was the way that
So that scientist, Dr. Gillespie, who's now becoming one of the best partners of becoming part of it. She's becoming the department head of biochemistry. So she studies signals perceive and respond to signals from their environment. And she works with a set of molecules that she thinks are like energy sensors. You can imagine with a plant, an organism that's making its living off of light, that it needs to modulate. If we ramp everything up too much, we have to modulate the output. So it's going to always be evaluating the quality and quantity of light being received by the plant and making adjustments to maximize that input. Okay? So her hypothesis was that protein was part of the energy sensing network, and they didn't respond. That's why it was so intriguing to her. Taking away that gene, knocking actually it was 2 genes, knocking them out meant the plant was so blind. They didn't notice the change, like a regular wild type. That make sense? Anything else?
All right, guys. So hopefully, as you were listening, you got some ideas, and you hopefully were able to brainstorm a little bit before you got here on the type of variables you might want to be using. What we want to do is split you guys up into some smaller groups and have you come up with a list of a few, thinking that we can then go back to our class groups, talk about those a little bit more, and come up with some idea, a list of maybe 2, maybe 3 things you might want to be testing. And while Mr. Lally's here, he can offer a little bit of input, tell you right away if that's something that might not be feasible. Just keep in mind the big point, it needs to be something relevant to the plant. People get excited about certain chemicals and things like that, and it's not something that they would encounter naturally, so we try to avoid that. So is that we have the laptops ready to use?
Be ready to be very quick.
All right, so what we need to do, and if the other teachers could help, we I think we said about 5 to 6 people per group. We're going to have one person who's the one that's kind of going to get the computer and be in charge of that. And here's what we want from you. Again, for each group, maybe 2 things or 1 really good one, 2 to 3 if you're like iffy on what is a variable we could be testing, and then maybe just like a brief statement on why that would be significant, why you think it would have something to do with, I guess, the effects of flavonoids or anything like that. So that's the type of thing we're looking for right now. If we have time, we might even have a representative kind of stand up and say, here's some about. The goal is by the time you guys leave here today, you at least have a good idea of what you might want to explore. It doesn't have to be set in stone, although if you're doing anything that has to do with the soil, it would have to be ready to go by Monday. So it's like that type of thing. We're going to want to know by Monday probably what we want to do for sure. So again, if like the teachers, if you want to get groups about 6 or so, and then I guess 1, how many people?
Yeah, no more than 3 computers per class, because apparently these kills the network.
Okay.
After 8 to 9 graders.
So we're on it.
Yeah, we have 18, 3 sets of 6.
Oh, okay, yeah, that works.
We have like 16.
Hey guys, don't come up for computers yet. Get to your
I'll go up there to get ready to get our computer.
I'll try
Okay. You just need to stand up.
The struggle is real. Recording? Oh, it says stop.
Just so everyone knows, hopefully y'all like watch this video sometime. I'm learning a lot about Arabidopsis today. I like the research. I handed in a notebook and commercial phones. The computer is still broadcasting. Still recording the video. So I figured I might as well get my little piece of input in there. I was asking a lot of questions today. That was Brittany. She was just talking. Patient. Talk to you later,
All right, folks. So now that you are getting your computers, I'm thinking three things we're looking for the variables. First off, what is the variable? Second off, why does it actually pertain to these plants? You may want to even get specific in why does this pertain to Arabidopsis. You may want to look up a little bit about where it lives and the type of conditions it's more related. Maybe most important part is how does it tie in with these flavonoid chemicals and the many, many effects you already talked about today. So those are kind of the three things I'm thinking. What's the variable? How does it actually pertain to these plants? And is there any tie-in with these chemicals that we're learning about now? Do some quick research. Just Google it. See what you can find. So if you can't find anything now, it's not going to get any easier when you try to write your paper. You know what I mean? So again, we're trying to get about two good things that maybe we can narrow down this plan.
I know what you
She already almost took theirs.
like for us to bring it up there.
What are you doing?
Smell it. Where's it, guys?